All right, welcome back, friends. Happy Tuesday. Tomorrow we go to lab and we do fun stuff together. Let's talk about today's concepts, and hopefully that will support and enrich your understanding of the lab and vice versa. I warned you I would do this. I threatened you I would do this. I promised you I would do this. So I'm going to do some cold calling based on the respiration diagrams that I assigned as homework. But remember, as always, this is not graded. You don't have to have the right answer. You don't have to give any answer. If you don't feel like it, you can just pass. That is also fine. It is kindness to all if you are not up to it and want to open the door for others to feel the same way. So with all that said, let's begin. So what I'm going to do is, here's sort of some things that happen during eukaryotic aerobic respiration. And we are focusing in this moment on eukaryotic cell. I'm going to ask you where these are happening. Let's begin with tau. Glycolysis in a eukaryotic cell happens where? I'm going to open my chat box up too in case I missed. Oh, where in the eukaryotic cell is this process of glycolysis taking place? If you can unmute, go ahead. But if you're not able to unmute and put it into the chat, that's also fine. If you want to pass, that's fine. Okay, so Tao said the membrane. That is not where it's happening. That is an excellent response. Thank you for giving me that. Let's go to Surmeet. Surmeet, what would you say? Where is glycolysis taking place?
Cytoplasm.
It is in the cytoplasm. Good job. Thank you both for that. Okay, next we're going to go to glycolysis, of course. We take glucose, we break it into two parts. That produces pyruvate. Pyruvate has to be oxidized to acetyl and then enter the Krebs cycle. Where is this happening? Sarah.
In the mitochondrial matrix.
That's right. In the mitochondrial matrix, which means that pyruvate, the product of glycolysis, has to travel across two membranes to get where it needs to go next in order to be processed prior to entering the Krebs cycle. In the Krebs cycle, it's basically pulverized down to carbon dioxide. And what we're doing is pulling all the electrons out of it. So out of what used to be glucose, we now have a pile of carbon dioxide waste, and a bunch of electrons have been put onto NADH. All those electrons go to the electron transport chain. They drop their cargo. They flow through the electron transport chain. Where is the electron transport chain located, Nikki?
I'm not too sure. The mitochondria.
It is the mitochondria. Let's see if anyone can be a little more specific, because it's a particular part of the mitochondria, but that's an excellent start. Thank you for doing that. Megan, where in the mitochondria is the electron transport chain?
I think the inner membrane.
The inner membrane, yes. There's an outer membrane and an inner membrane. It is the inner membrane. Good job. All right. Now, what happens is, of course, as the electrons flow through the electron transport chain, their energy is siphoned off and used by the members of the chain to pump protons across that inner mitochondrial membrane. Where do those protons end up after they've been actively transported, forming an acidic region, also known as a hydrogen ion gradient, also known as a proton gradient? Where is that part of the cell? What compartment is becoming acidified? Lori, what would you say?
I'm gonna pass.
That is absolutely fine. And thank you for doing that. Now everyone else can sort of breathe a sigh of relief. That is very kind. Let's move on and ask Kelly if she knows.
Is it in the intermembrane space?
It is in the space in between the outer and inner membrane of the mitochondria, a nice safe place cordoned off from everywhere else. So you're not acidifying the matrix or the cytoplasm, but a part of the mitochondrion and the cell in general that is not used for anything else. Good. Okay. What happens next is that hydrogen ion gradient is allowed to diffuse back through ATP synthase. It's a flow, it's kinetic energy. That flow of protons causes the ATP synthase turbines to actually literally spin, creating kinetic energy. And that kinetic energy is used to be converted back into chemical energy. It's used to make ATP. And so ATP synthase itself is located where, Caddy, where in the cell is ATP synthase, the machine that is letting those protons come back into the matrix from the intermembrane space? Where is this ATP synthase machine? If you know Caddy, go ahead and give us your best idea. The inner mitochondrial membrane, that's right. Okay. And so those protons do end up back in the matrix, but meanwhile, there's still this gradient. As long as we have electron transport chain is functional, we still have more hydrogen ions on one side than the other. So we have a continual spinning of ATP synthase, generation of ATP. And ultimately in a eukaryotic cell, let's have everyone write this into the chat. How many ATPs do we get, guys? Fabulous. Yes, it is 36. Okay, so last time we briefly compared this to prokaryotic cells, and I've done this several times this quarter. Let's see what we remember. No pressure, just do your best. You can always pass. You can always guess. You can be wrong. I don't care. Just, play the game. So let's go to John. Where's glycolysis happening in a prokaryotic cell? John says cytoplasm. He is correct. Janelle, pyruvate oxidation in the Krebs cycle. When the pyruvate outcome of glycolysis has to be modified and then fed into the Krebs cycle, where is all that happening in a prokaryotic cell, Janelle?
Cytoplasm.
Also the cytoplasm. Good. The electron transport chain is located where in a prokaryotic cell, Haley?
That's the plasma membrane.
That is the plasma membrane. What is the region that is where the hydrogen ion gradient forms in a prokaryotic cell, Ayumi?
Extracellular space.
That's right. Those protons are pumped outside the cell, and then they're just like hanging around in a cloud of acidicness around the near the membrane. And then ATP synthase located where, Amy?
That would be the cell membrane.
You're absolutely right. Good job.
Or the plasma membrane.
Yes, cell membrane, plasma membrane, basically the same as far as our class is concerned. So good work. All right, and everyone, what is this answer? ATP per glucose in a prokaryotic cell is how many? Put that into the chat. Perfect. So what was the reason that we have 2 extra ATPs in a prokaryotic cell, a less complicated cell, a smaller cell, one you might imagine is not as good at doing what it's doing, maybe a more ancient, so yet somehow it's doing a better job of getting ATP out? What's the reason for that? Raise your hand if you know or want to speculate. Go ahead, Sarah.
Because they need to use the extra ATP to travel into the mitochondria for eukaryotes.
Yeah. Big difference is right here. End product of glycolysis is pyruvate. needs to get into the mitochondrial matrix, has to cross the outer and inner mitochondrial membrane to do so. That costs 2 ATPs. Don't have to do that in a prokaryotic cell. So the prokaryote just gets to keep all the ATP it's made. The eukaryote has to spend a bit. Okay, so I want you to have a look at your diagram right now. And I showed you this before. I'm just going to really briefly review it again. Obviously, I'm not an artist. This is pretty low quality, I guess. But what I want you to think about is that you are able to do this kind of in your head, kind of imagining where's stuff happening. In a prokaryotic cell, we've got glycolysis producing pyruvate that enters the Krebs cycle that yields electrons that are being removed. Those removed electrons on NADH are then oxidized back to NAD+. The electrons flow through the electron transport chain. The energy from those electrons is used to generate this extracellular hydrogen ion gradient that comes back through ATP synthase to generate energy for the cell. And in comparison to that, in a eukaryotic cell, we have the inner and outer membrane of the mitochondrion, the cytoplasm, the matrix. Glycolysis is happening in the cytoplasm. We get pyruvate, we import it across the 2 membranes of the mitochondrion to end up in the matrix. And at that point, we modify it, we send it to the Krebs cycle, and we extract as many electrons as we can, making NADH. And then just like in this cell, the NADH is dropping its electrons off at the membrane. And those electrons go through the electron transport chain. Their energy is used to pump protons into the intermembrane space. The protons end up on oxygen producing water. They come back, the protons come back through ATP synthase producing ATP. So you can see what I'm not showing, what I don't expect you to remember, which is all of the 11 steps in glycolysis and all their intermediates and their enzyme names, or all of the different steps in the Krebs cycle and all their enzyme names and intermediates, and none of the really specific details or a lot of accounting as to exactly when and how many things happen, but just the overall picture and kind of where stuff is happening. All of this is in service of allowing us to understand the major critical differences between the different types of metabolic strategies that organisms employ in order to survive in the microbial world. So if we think about the 2 forms of respiration, and we briefly reviewed this last time, aerobic versus anaerobic, what is similar between all forms of respiration? What pathways do they both rely on, or what would you say is like similar between these pathways? There's not just one right answer to this, but what would you say? Put that into the chat. Okay, they both use glycolysis at the beginning. That's right. So we start with glucose. We go through glycolysis, we break it into pyruvates. They both produce ATP. Yes, that's correct. Anything else that comes to mind as being similar? Any pathways they share in common besides glycolysis? They both use the Krebs and electron transport chain. Yep. So they go through this same process, break glucose down, extract the electrons, use the energy from electrons to make ATP. The major difference between them, of course, is what? The terminal electron acceptor is oxygen or something similar but different from oxygen. And so aerobic respiration uses oxygen as the terminal electron acceptor at the end of the electron transport chain. And anaerobic respiration uses something else. It's always going to be something inorganic and similar to oxygen in that it is electronegative and has similar chemical properties, but is never as electronegative. Nitrate, sulfate, carbonate would be typical examples. Because of the differences in the chemical nature of whatever this alternative is, anaerobic respiration always has a lower ATP yield than oxygen and aerobic. So here we have a diagram in a prokaryotic cell. We're focusing on prokaryotes in this class, and this is what this is, right? We've got glycolysis producing pyruvate. We've got the Krebs cycle producing NADH. We've got oxidation of NADH to put those electrons into the electron transport chain. We're using that energy to pump protons outside the cell. They're coming back through ATP synthase and making ATP. Where do those electrons go at the end of the electron transport chain? In aerobic respiration. Everyone should answer this. We basically have this molecule here, glucose with the electrons in it. We go through a whole bunch of steps to remove those electrons, put them through the electron transport chain to extract their energy. And then the electrons go where? At the end. That's the terminal electron acceptor in aerobic respiration. Yeah, exactly. Oxygen. That produces water. This is aerobic respiration. This is how it works in our bodies. This is how it works in bacteria that do this. Now, if there's no oxygen and we want to still do respiration, if we have the right tools, we can do anaerobic respiration instead. So would we still use the same enzyme? Would this enzyme that's specific for oxygen be able to participate in anaerobic respiration? Your instincts are correct. This is very specific. It specifically puts electrons onto oxygen. Oxygen is a substrate. It can't use other things. So if this enzyme complex is all you have the genes for, then this is your only option. And if oxygen goes away and you rely on this to make energy, you will die. But some organisms have an alternative, a different thing. Let's say, just for example, nitrate reductase, though this is not the only option. If you had nitrate reductase, you could put electrons onto nitrate instead and produce nitrite. Now you can do anaerobic respiration, at least this form of it. Okay, now you might be a different organism and you don't have nitrate reductase, but maybe you have carbonate reductase, or maybe you have sulfate reductase, or you have something else that can use a different substrate that's similar to, but not identical to oxygen. If you have the genes to do that, you have the ability to do anaerobic respiration. Now, I've said this already. First, let's find out if you remembered it, and then I will try to explain it. Aerobic ATP yield, how much ATP you get in aerobic respiration from one glucose, in a prokaryotic cell is 38, right? If we do, let's say, nitrate reduction or carbonate or sulfate reduction, if we do a different type of reduction, we don't use oxygen as our terminal electron acceptor. Do we get 38 ATP? Do we get more? Do we get less? If you're unsure, put in a question mark. If you want to speculate wildly, go ahead. If you want to roll the dice, you've got 50-50 chance of being right. I do want you to answer this question. Great. Okay. So the answer, if at all else fails, you'll need to memorize this, but I hope that I can tell you why, okay? The answer is less, and many of you put that. And some of you were like, I know it's supposed to be less, but I don't know why. And some of you were like, it is less. So just very briefly, two things that may explain this for you. Oxygen is more electronegative than any of the alternatives, which means it pulls on or accepts electrons more readily. And that's necessary if you want really exhausted, low energy electrons to actually go to a molecule. They have to be more electronegative than the thing before. It turns out that these other alternatives, these other enzyme complexes like nitrate reductase, carbonate reductase, sulfate reductase, the substrate they use is not able to accept electrons that are as tired. And so there isn't as much pumping of protons, using up the energy of the electrons, and not as much pumping of protons. So this proton gradient is not as strong, and we can't get as much ATP. So I hope that's enough to sort of convince you. And if you can't be convinced by that, then just memorize it. Okay, next we're going to compare these two processes, respiration and fermentation. These also share some metabolic features. Some aspects of these two processes are in fact the same. What pathway or pathways do they share in common? Yes, they both start with glucose and they both break it in half to produce pyruvate. That's where things change. What happens next to pyruvate in respiration is that it is changed to acetyl and then gets into the Krebs cycle. But in fermentation, that does not happen. So we don't get the Krebs cycle. We don't get the electron transport chain, and we don't make any more ATP after glycolysis. And so what does that tell you about all forms of fermentation ever in terms of ATP yield? How much ATP does fermentation give you, regardless of whether it's lactic acid or ethanol or xylose or mannitol or X, Y, or Z fermentation? You always get how many? That's right. You always get the amount you get from glycolysis. And so in glycolysis, you made 2 ATPs, and that's what you get from every fermentation form. The other difference is, what is the terminal electron acceptor, right? So this may be an odd concept because, of course, in respiration, it's more sensible to talk about this. There's an electron transport chain, the electrons are going through it and they come out the end and they have to go somewhere. That's the terminal electron acceptor. But fermentation has one also, because in the process of doing glycolysis, you do take some electrons out of glucose. Now they're on NADH. They still have to go somewhere because NADH is an intermediate, it's not an endpoint. So they still have to go somewhere. They get dumped onto an organic molecule. So respiration, it's an inorganic molecule like oxygen, sulfate, carbonate, nitrate, etc. And in fermentation, it's an organic molecule like pyruvate. So all forms of fermentation, 2 ATPs. All right, so let's do a little diagram here. So here we've got a little cartoon of respiration without any reference to cell structures now, just what's the biochemistry. Glucose broken in half to 2 pyruvates. Along the way, we take out some electrons. Pyruvate converted to acetyl. Along the way, we take out some electrons. Enters the Krebs cycle. Along the way, we take out some electrons. Those electrons go to the electron transport chain and are used to make ATP. And then these empty NADHs run back and gather more electrons. So that's respiration. And what we can see is that at the end of the electron transport chain, our terminal electron acceptor is going to be whatever it is. If it's aerobic respiration, it's going to be oxygen. If it's anaerobic, it's going to be something oxygen-esque, but not oxygen. But fermentation ends right about here usually. Or this is where the similarity ends, I should say. So we still have glucose being broken in half. We still have some electrons being removed and put onto NAD+, forming NADH. And this is necessary for this part to happen and for ATP to be made. But now we've got an NADH running around, and we need it to be turned back into an NAD+ just so it'll be able to work in glycolysis. So we have to drop our electrons somewhere, and a simple way to do this is just drop them right onto pyruvate, and that turns it into lactic acid. Pyruvate's a 3-carbon molecule, add some electrons, you get a 3-carbon lactic acid. This is known as lactic acid fermentation. And so what you'll notice is that the electrons that were removed from glucose in fermentation were never used to make any ATP. The only ATP is made just during the regular normal glycolysis reactions. Is this the only type of fermentation that exists in the world? It's certainly what happens in our bodies. This is the only type of fermentation that happens. Yes or no, guys? What do you think? If you absolutely don't want to answer yes, no, or question mark, put in kittens. I like kittens. Great. So this is certainly what happens in our bodies. And there are some microbes that do this and only this. But there are many, many types of fermentation that can happen. And I'll just give you one other example, though it is not the only one, but it is the only other one you're required to know much about. And I'm presuming most of you are aware that wine and beer comes from the metabolic activities of yeast. So that produces alcohol, a 2-carbon molecule, and gas in the form of CO2, which is achieved by doing this. We do glycolysis, and then we let the next stage in respiration happen, actually. We remove one of the carbons, throw it away as CO2, there's where the gas happens. And then we reduce, we add our electrons from NADH to this 2-carbon molecule and turn it into ethanol. But this is not the only type of fermentation. There's all kinds of variations on this theme. And this arrow, this biochemical reaction, doesn't happen just spontaneously. What is responsible for changing, let's say, pyruvate into lactic acid or changing acetyl into ethanol? What's responsible for that? How does this take place? Is this just you just like let it sit around until it magically happens? Or is there something that's in charge of this? How do chemical reactions happen in living systems? What drives them? That's right, Kelly. Exactly. Enzymes do. Enzymes are proteins. proteins are encoded by your DNA. So that means that what determines what type of fermentation you can do, if any, is what your genes say you can do. What enzymes do you have in your genes? That's what matters. So that's why you can do lactic acid fermentation, but not ethanol fermentation. And that's why yeast can do ethanol fermentation and not lactic acid fermentation. And that's why bacteria, if they can do lactic acid fermentation, do that. Or if they do something else, they do something else. Okay. This picture is right out of your video lectures. It nicely summarizes in a better way than my silly diagrams the difference between respiration and fermentation and where they kind of diverge. But I think this is also important to notice, and you don't have to memorize this. You have to understand the concept of this. That pyruvate is the end product of glycolysis, and then what enzymes you have in your genetic makeup determine what happens to that pyruvate. If it's going to be fermented, or if fermentation is going to happen, the fermentation products are determined by what enzymes you have. So here is one version of fermentation. That's something we can do, and what microbes can do that too that you've heard of. Many species of bacilli, lactobacillus, and streptococcus. We know our good friend Saccharomyces cerevisiae. This is the budding yeast that makes beer, wine, and bread. Has different enzymes and can produce ethanol and carbon dioxide. My personal favorite, the Swiss cheese organism, Propionibacteria, produces propionic acid, giving Swiss cheese that tangy flavor that's so distinctive. And we can see a variety of other options. I want to take a second to talk about the catabolism of other macros besides carbohydrates. We've talked about glucose and other carbs, both in prokaryotes and in our bodies, get converted into glucose so that they can be fed into that pathway we've just talked about. But if we look just at respiration, let's talk briefly about where in respiration things like proteins and lipids come in, because those are also something that commonly organisms can eat. We can, for example, and many bacteria also, not all of them, but some of them, most of them, are able to use lipids and proteins as a source of energy. So here we've got, again, another view of respiration. Glucose broken down to pyruvate and glycolysis, changed to acetyl in the intermediate step, fed into the Krebs cycle, and then the electrons that are recovered are sent to the electron transport chain to generate ATP. And what I'm abstracting here is just the number of carbons of the molecules that are in here. And I want to point out that, of course, glucose is a 6-carbon molecule. Pyruvate is a 3-carbon molecule. Acetyl is a 2-carbon molecule. And then in the Krebs cycle, oxaloacetate right here is a 4-carbon molecule. When you add acetyl to it, you form a 6-carbon molecule, which is citrate. And then you subsequently remove a carbon, creating a 5-carbon intermediate, remove a carbon, restoring the 4-carbon form. And this is going to be important when we think about where these other macros come in, because if you know anything about biochemistry or nutrition, then you may remember what a triglyceride or a fat looks like. Typically in our diets, like when we eat a sandwich and it has fat in it, maybe the butter, or oil in the mayonnaise, or when a microbe is swimming through, a bacteria is swimming through the world and it finds some food, if it's eating lipids, the lipids are in the form of triglycerides because that's the storage form in bodies, right? So triglycerides are a 3-carbon backbone and some fatty acid tails. Fatty acids are really long chains of carbons. There's also hydrogens and oxygens in this molecule that I have not diagrammed. And remember that triglycerides are like 3 fatty acid chains off of this glycerol, whereas phospholipids are 2 with a phosphate group. So what happens to lipids is really fascinating. First of all, the legs are chopped off of the 3-legged stool. These fatty acids are released. The fatty acids are always an even number of carbons. Most common in the human body is 16 carbons or 18 carbons, but there is a variety of them, but they're always an even number. What happens next is that the 3-carbon glycerol gets fed into respiration around the level of pyruvate, which makes sense because pyruvate has 3 carbons and glycerol has 3 carbons. And then these long chain fatty acids are chopped into 2-carbon units. that are fed into this level at the acetyl level right before the Krebs cycle. So this is relatively straightforward. Now, if we look at what a protein is, it's a polymer of subunits, and each subunit is what? That's being diagrammed over here. Proteins are made of a series of covalently bonded what? Proteins are a long chain of not repeating subunits, but various linked monomers. And what are the monomers in protein? That's right, Sarah. Amino acids. So remember, we've got a long chain of amino acids, and each individual amino acid kind of has this structure: a central carbon attached to a carboxylic acid group attached to an amino group. The first thing we have to do, and let me just see if I can get this to back up. There we go. Is we've got to, right? We chop these amino acids so they're separate from each other. And then we actually deaminate, remove, lop off, chop off the amino group and throw it away. NH2, which very quickly becomes NH4, nitrogen attracts hydrogens. NH4 is ammonia. That's really dangerous. So in our bodies, we convert it quickly into urea and it gets excreted in the urine. But organisms that are single cell prokaryotes would just chop it off and literally throw it into the environment. This is alkaline. It causes the environment to become alkaline. Then what's left is the carbon backbone of the amino acid. And it's somewhere between 2 and more than 2 carbons, depending on the R group, what's up here, right? Could be just a hydrogen here, in which cases it's 2 carbons. Could be a methyl group, which is 3 carbons. It could be longer, could be 4, 5, or 6 carbons. But where the amino acids go into respiration is determined by how many carbons there are. If it's 3 carbons, it goes in around the same place glycerol does. 2 carbons go into the acetyl level. And then 4, 5, and 6 can go into different parts of the Krebs cycle. So it's more complicated, protein metabolism is. One other thing I'll mention, and I don't know if you're going to remember this long enough for it to be helpful, but I hope so. You may have a vague recollection of some of the other functional groups or R groups of amino acids, like, for example, tryptophan, which has a rather unusual, like a benzene ring. It's not straightforward like this kind of structure. It's like a whole circle of carbons and basically cannot be metabolized by most organisms. So when you have a tryptophan, and there's a few other amino acids like this, you need a special enzyme to chop the special R group off of the tryptophan and use the rest of the molecule for energy. And if you don't have it, you can't use tryptophan. It just ends up not being digested. So I'm mentioning that because a lot of you are going to end up doing something called the indole test. The indole test tests for your ability to remove that little ring from the tryptophan amino acid to use it as energy. So there's lots of applications of this type of biochemistry to your unknown ID project. Okay, so the next topic we have to tackle is exoenzymes. Very important to prokaryotic cells. Basically, exoenzymes are enzymes that are sent outside the cell to digest the environment. So think about why that's important, right? We don't use these in that same way. I suppose you could argue that we do If you want to think about your GI tract as not being really part of you, but actually part of the environment, it's true. But we have a GI tract where digestion takes place, but bacteria don't. And they're also prokaryotic, not eukaryotic, which means that let's suppose that there's bacteria swimming through the world and it comes across a molecule of starch, a long chain of glucoses. Beautiful starch, lots of energy there. It can't engulf that starch and bring it inside and digest it inside the cytoplasm because it doesn't have the ability to do endocytosis. It's a prokaryotic cell. If it wants to bring things in across the membrane, it can bring them in one small monomer at a time. It could bring in one glucose. It could bring in a disaccharide, but certainly not a long chain of glucoses like a starch molecule. Impossible. So what's the solution? It secretes an enzyme outside the cell to chop the starch into small pieces, and then those pieces can be imported one by one. So what is that enzyme that bacteria use, which by the way is the same one that you use to break starch, a carbohydrate produced by photosynthetic organisms, primarily plants? What is it? What is the enzyme that breaks starch down into the glucose subunits that bacteria use and that we also do? It's in your saliva. It's why when you eat a cracker, a starchy cracker, when you just chew on it long enough, if you leave it in your mouth, it'll start to taste sweet. What is that enzyme called? That's close, Meliana. That is a different enzyme that also actually is made in the mouth. So it's not lipase. Lipase digests something else. Yes, John. Yes, Meliana. If you took nutrition, you will have covered this. Maybe you covered it in A&P. I don't know. It's amylase. So we have it in our mouth, and we also have it in our small intestine. The pancreas makes it. So our saliva, it's in our saliva, our salivary glands make it. And the pancreas also makes it and dumps it into the small intestine. Bacteria secrete it into their environment. Their environment is their GI tract. Okay. So think about your body again. When you eat a sandwich, let's say, and it has butter on it, it's full of triglycerides, or you eat, pasta with the olive oil on it, the oil has triglycerides in it. That's our lipids in our diet. We need to break those triglycerides down to fatty acids by chopping them off of the glycerol backbone. What is the enzyme that does this? It's already been mentioned. Yes. Yes, exactly. So we're trying to break lipids basically. And so we have lipase. Now we have a little bit of we have a little bit of lingual lipase, a little bit more when we're babies, but just a smidgen. So we can start to digest fast in our mouth. Mostly it starts in the stomach. We have a lot of gastric lipase, and then there's pancreatic lipase as well added in the small intestine. Bacteria don't have any of those things, so they just spew it into the environment if they've got it to digest triglycerides so that the fatty acids are liberated. And then the fatty acids can come across the membrane. And last but not least, we also need to break down proteins. So what happens is this does not happen in our mouth. We swallow the proteins into the stomach, which is acidic. The acid causes the proteins to denature, relax into long limp noodles, long chains of amino acids, and then an enzyme made in the stomach chops the protein in between the amino acids. What is the name of that category of protein, enzyme that digests protein into amino acids? Yeah. So we have proteases that are made in the stomach. And that's not sufficient to get the job done. It gets a start on it. And then there's more proteases in the small intestine. Again, bacteria don't have a GI tract, so they just send the protease out into their environment. Okay. Our next topic is going to be looking at the carbon fixation pathways, but I think this is kind of a natural break. So we're going to take our 10 minutes. We're going to come back at 5:55 and talk about those other ATP producing, but also sugar producing pathways. I will see you in 10. Okay, welcome back. We're going to do our favorite game, which is cold calling. And this is a part of metabolism that I am aware is not as commonly covered in previous courses. So if you don't remember from the prep that you did, or didn't know this coming into this class and are just sort of having a mind blank, that's understandable. Again, all you need to do is pass, no harm, no foul. However, let's see if we can get some answers to some questions. So again, we're going to focus on the eukaryotic cell first, which is the one, if you've covered photosynthesis in the past, is most likely the version that you covered, and ask where stuff is happening. So photosynthesis is basically a two-part process. The light reactions that involve protein-based photosystems, like a little solar panel to collect sunlight, that energizes electrons that are stolen from water and feeds them to nearby electron transport chains that squeeze the energy out of the electrons. So the electrons are taken from water, energized by the sun, and then those energized electrons are fed directly into an electron transport chain, which can harvest the energy and use it to pump protons across a membrane, establishing a proton gradient, which allows you to generate ATP through ATP synthase. Then there's a second step we'll talk about in a second, but those are the light reactions, the goal of which are to generate ATP and electrons. Where are the photosystems and their associated electron transport chains located in a eukaryotic cell? And let's start with Amy, if you know or can guess.
I'll pass.
No problem. Thank you for doing that. Next, we're going to ask Tao, if you know. And I think last time Tao had to put her answers into the chat. Let's see if she's there. Are you able to answer us? I feel like this is a seance, right? Are you there? Okay, let's move on. The next person I'm going to ask is Ayumi. Where are the machines? Oh, Tao says she'll pass you. Thank you for answering that. I appreciate you. Okay, photosystems and electron transport chains. It's the same location. Where is it, Ayumi, in a eukaryotic cell?
Thylakoid membrane.
That's right, in the thylakoid membrane. Shortly, we're going to have a diagram, so you'll see what that is if you're feeling a little shaky on what it is. But the thylakoid membrane is the inner membrane system of the chloroplast in eukaryotes, which is where photosynthesis happens. Now, the purpose of this is to establish a proton gradient, right? To pump a bunch of protons onto one side of a membrane so that they're at high concentration, and they'll let them come back through ATP synthase to generate ATP. Where is that hydrogen ion gradient forming? Surmeet.
For ATP formation?
Well, on the way, sort of on the way to ATP formation, we have to put a whole bunch of hydrogen ions into one place so that they're at a high level of concentration. Where is it that they are accumulating?
I'll guess, I'm not sure. Maybe it's if stroma.
That's a great guess. It's not right, but it is close. Okay, so what Surmit's talking about is that the thylakoid membrane is a membrane that divides the stroma on one side from the interior of the thylakoid on the other. So it's actually the other side of the membrane. The interior of the thylakoid is where it's happening. Okay, good job. Now, what about the dark reactions? So the whole point of this light reactions is to make a bunch of ATP and also to make some energized electrons. We're going to use the energy and the energetic electrons to knit CO2s together into glucose in the Calvin cycle. These so-called dark reactions don't rely on darkness. They just don't need light. But where did these take place? Sarah.
The stroma of the chloroplast.
That's right, in the stroma of the chloroplast. And if you're wondering what that is, guys, we're going to have a diagram in a second, so hang tight. But it is the fluid-filled interior of the chloroplast that is functionally equivalent to the fluid-filled interior of the mitochondria known as the matrix. So this is sort of like the chloroplast matrix. Okay, so in a eukaryotic cell, there's a membrane called the thylakoid membrane where we put the machines that are responsible for the light reactions, including the electron transport chains. And then in the stroma, which is the adjacent fluid, there's the Calvin cycle happening to do the dark reactions. And in order to accomplish the goal of the light reactions, we have to make a hydrogen ion gradient, which is basically an acidic compartment in the inside of the thylakoid. Thylakoids are little pouches. So where are these equivalent things happen in prokaryotes is a question, right? Because prokaryotes can do all this stuff, but they don't have any of these structures. They don't have a thylakoid membrane. They don't have a stroma because they don't have chloroplasts. So if the light reactions have to take place in a membrane, where in the prokaryotic cell are they happening? Nikki. Nikki's going to pass. Excellent. Miliana, do you have an answer for that? Or a guess? She's not sure. Okay, no problem. Let's go to Haley. What about you? What do you think?
I said plasma membrane.
Yes, you would be correct. And all of these other things are happening in the equivalent places, like the cytoplasm is where the Calvin cycle is happening. And the extracellular space is where the hydrogen ion gradient is forming. And this should sound really familiar because that's also where the hydrogen ion gradient is generated during respiration in bacteria as well. Good job, guys. All right, so I'm going to, again, draw you some little cartoons. And this is kind of the level of understanding I want you to have. So draw these cartoons yourself until you feel confident. Let's start with the eukaryotic cell. It's got a chloroplast in it. The chloroplast has two membranes, outer, inner, and then a third set of membranes, which is going to be shown here as a big pouch called the thylakoid, and it's got a membrane around an interior. There's the photosystem energizing electrons from water using the sun. That's feeding it through the electron transport chain, using the energy from those electrons to create an acidic compartment on the inside. More sun re-energizes those same electrons, so they end up on this electron carrier. The protons come back through ATP synthesis to generate energy, and ATPH brings electrons to the Calvin cycle shown right here. And so we get 3 carbon dioxides plus all this ATP plus the electrons from NADPH put through a rather complex cycle to create a 3-carbon sugar, 2 of which get stuck together to make 6-carbon glucose. Here's the thylakoid membrane. Here's the interior of the thylakoid. Here is the stroma where the Calvin cycle is happening. If we look at a prokaryotic cell, all the same things happen, but we just have fewer places for things to happen. So it's much simpler in a sense. The photosystems are not on the thylakoid membrane, they're on the plasma membrane. We have water being energized by the sun, fed through an electron transport chain. The energy from those electrons is used to pump protons outside the cell. The electrons are re-energized by the sun and put onto NADPH for later use during the carbon fixation part. ATP is generated by allowing all these protons to come back through ATP synthase. Now we've got ATP in the cytoplasm, high energy electrons, that comes together with carbon dioxide in the Calvin cycle, also happening in the cytoplasm to produce 3 carbon sugars, which are stuck together into glucoses. There's lots of stuff you could know about the Calvin cycle, but I'm not going to require you to know that. I just want you to basically understand that what it is doing is it's taking electrons, energy, and carbon dioxides and smushing them all together to make a 3-carbon organic molecule, 2 of which are required to form 1 glucose. All right. So this part might seem kind of heavy to you. I'm giving you an aspirational goal to understand this eventually. You want biochemistry to not just be about all the picky details in this arrow that you could learn and memorize, though that may be important at some point, but rather you want to understand conceptually what's happening. What's happening is that carbon dioxide is being reduced. And what does reduction mean, guys? Put that into the chat. To reduce a molecule means to do what to it? Remember our redox conversation from last class. When we reduce something, we do what to it? That's right, Sarah. That's right, John. When we reduce something, we add electrons to it, and electrons are mostly energy. So when we do that, we are creating a molecule that's higher energy, right? This is really critical. Having organisms sort of at the bottom of the food chain, so to speak, doing this is really important to generate the organic material that the rest of living systems depend on. Because let me ask you this question real quick. Can you do this? Can your body do this? Can you take carbon dioxides and reduce them to organic compounds? Yes or no? That's right. We can't. Macroorganisms, eukaryotic macroorganisms typically can't, and a lot of bacteria can't either. So those brave few carbon reducing organisms are absolutely fundamental to allow life to continue to exist. Okay, so if we understand respiration and photosynthesis at this kind of high level, then it will make it easier to understand these kinds of comparisons. Okay, so first we can just kind of ask this question, like where's the Calvin cycle and Krebs cycle taking place in these 2 different cell types? In eukaryotes, we have the Calvin cycle taking place in the stroma and the Krebs cycle taking place in the matrix. These are both fluids on the interior of an organelle around pH of 8, where chemistry can occur. In a prokaryotic cell, both of these happen in the cytoplasm. Are these pathways overall oxidizing or reducing with respect to the carbon molecules that are going through it? We already kind of addressed this when we were talking about the Krebs cycle because it's part of respiration, right? Respiration overall, we decided in terms of glucose is oxidizing because we start with glucose and remove electrons from it, right? So we're oxidizing the glucose. What happens in the Calvin cycle is we take carbon dioxide and we stuff electrons onto it. So is the Calvin cycle overall reducing or oxidizing with respect to those carbon dioxides and the sugars that are produced? Is the Calvin cycle in photosynthesis overall oxidizing or reducing? Pick one or put a question mark if you don't know. So what we're doing is we're adding electrons in the Calvin cycle to carbon dioxide. That is the definition of reducing something. We're reducing it. We are adding energy to it. We are adding electrons to it. We're creating something higher energy. The Krebs cycle is the opposite. We're removing electrons from acetyl. And that is oxidizing it. That is reducing its energy. So in the Calvin cycle, carbon dioxide is reduced to, has electrons added to it, is reduced to an organic molecule. And in the Krebs cycle, an organic molecule is oxidized to CO2. When we remove those electrons, we also break the bonds in between the carbon-carbon backbone of the acetyl and throw away the CO2 as waste. So big picture purpose of the Calvin cycle is carbon fixation, creating glucose for the cell and really for all living systems. And the Krebs cycle's goal is to take that glucose or the glucose derivative. By the time it gets to the Krebs cycle, it's acetyl, but that originally came from glucose, and burn it for energy. So photosynthesis is one way. Obviously plants do this, but also microbes, some bacteria do this, algae do this. So there's eukaryotic microbes that do photosynthesis, algae, and there's prokaryotic microbes that do photosynthesis, some bacteria. Is there any other carbon fixation pathway? And if so, what is it? Is there any other process which will take CO2s out of the air and turn them into sugar? All right, yes, Sarah is right. It's chemolithoautotrophy. And guys, remind me what, and have everybody contribute. What does this part mean, litho? If you don't remember, put in a question mark, but do answer. Yes, Tal, you're right. That's right. So this is a way of making energy out of rocks, basically. But what's really happening is we have some kind of inorganic molecule, usually like a metal ion, because metal is, I don't know how much chemistry you had or how much you remember, but metals are special because their electrons are relatively easy to remove. So maybe a metal ion that we can take an electron off of, feed it through an electron transport chain, use that to make a proton gradient that comes back through ATP synthase to generate a ton of ATP. And then we add that to CO2 plus some electrons, and we make a 3-carbon. Sugar, we put 2 of those together into glucose. And this is, of course, important because we need carbon fixation to happen, not just where there's sunlight, but also in areas of the world where there's living things that are living in the dark, like the bottom of the ocean, underground, in cave systems, etc., such as this location here. Okay, so now we get to talk about the really fun stuff. Okay, thanks, Tiffany. I was wondering who that was. I appreciate that. You're going to use this understanding and apply it in your unknown ID process. So when you look at the microtest reference, a pretty decent number of the tests that we have available for you are metabolic tests and really pretty relevant to what we've just talked about. Many of you had to do, for part 2, a thioglycolate in order to determine whether your organism is, let's say, an obligate aerobe or an obligate anaerobe or a facultative anaerobe or aerotolerant. So let's look at this thioglycolate test again. We talked about this during the growth section. We looked at the different patterns of growth, and we talked about what their classification is. Now let's talk about what's happening. So when you look at these tubes, can you tell which of them is definitely doing aerobic respiration? Put the letter or letters into the chat. Which of these tubes represents an organism that is capable of doing aerobic respiration? Great. Good job, everyone. Okay, so everyone pretty much said C. Look, because C is growing at the top. It must be doing aerobic respiration. That's true. There's one other one, and it is B. And then some of you guessed A. I suspect that's because of the trick of the light. This is not actually more growth. This is just shininess. So it's actually a very even pattern of growth all the way through. How do we know that B is also doing aerobic respiration? Anyone know what clued you in? Raise your hand. Some of you did just choose B and C. You recognize that this is doing aerobic respiration. How did you know? What clued you in? How did you know it was B and not A? Raise your hand if you can answer that. How did you know it was B and not A? Let's see who answered that. I'm going to ask Sarah. Can you tell us why you said B and not A? Which is correct, but how did you know?
Because the majority of the growth is congregated at the top. It looks like it's growing throughout, but it prefers to grow at the top, close to where
Yeah.
oxygen is.
Yeah, there's a much faster, denser growth at the top than there is at the bottom, which suggests that there's something different happening here when oxygen is present than is happening here when it's absent. And that's not the case for A, which is growing the same all the way up and down. Which of the tubes is capable of doing anaerobic metabolism? So there are three, and most of you have called them out. We know that there's anywhere we see that there's growth at the bottom, we know that it's capable of doing some kind of anaerobic metabolism. We don't know what it is. Is it anaerobic respiration? Is it fermentation? We can't be sure, but we know for sure it's doing some kind of anaerobic metabolism because it is alive down here. Now, can we be sure? Can we just by looking tell whether or not it's fermentation? Some organisms can do anaerobic respiration and not fermentation. So do we know who's doing what here? Just by looking at these tubes? Can we tell what form of anaerobic metabolism it is? You guys are absolutely right. It is not possible to know from looking, right? All we know is that it's alive. We don't know why it's alive. The only thing we can say for sure is we know that C is not doing fermentation because it is dead. If it was able to do fermentation, we would see some growth at the bottom. Okay, now in the growth section, we talked about which defense against oxygen enzymes these different categories of organisms have. Let's revisit that. So which of these have the enzyme SOD, superoxide dismutase? Just put your letters into the chat. Don't worry about being right. Just put something in that seems right to you. That will engage your brain and you'll remember whatever the answers are, if you were right or if you were partly right. Who's got SOD? I'm just going to wait for y'all to log your answers in. Again, don't be worried about being wrong. Just make sure you have a guess of some kind, and then we'll see how we do. Great. So just to remind you, SOD is that enzyme that detoxifies the worst reactive oxygen species, the superoxide anion. And if you grow in air, whether or not you use air in your metabolism or not, you must have this enzyme. So we can be confident that A, B, and C all have it. And we can also be confident that D doesn't, because otherwise it would be alive up here. Now, remember that catalase is another one of these defensive enzymes for organisms that can grow in air, but it's only found in organisms that actually use oxygen in their metabolism. And this is the tricky part, and catalase is not the only option. Peroxidase could be used instead. So can we be sure who here has, we already know that D does not have catalase. That's the one we can be sure of. Can we be sure that these guys do or don't have catalase? Given that there's an alternative to catalase. And I'll wait for a few more answers to trickle in, guys. Put in your votes. Can we be sure which of these have catalase? So we can speculate. We can know who definitely doesn't have it, but we can't be sure. So one thing we know is that this guy doesn't have it. And one thing you may have to memorize is that this aerotolerant guy doesn't have it. He doesn't use oxygen in his metabolism, so he doesn't generate enough oxygen species to really worry about this second type of defense. It has SOD and that's it. These might have catalase, but we can't be sure. This is actually useful because having catalase or not having catalase is frequently used to distinguish groups of microbes from each other, which is why catalase is another test along with thioglycolate that could be really useful in your part 2 unknown ID process. Another enzyme that you may test for is oxidase. And can we be sure who has oxidase? You might think so, right? We know that cytochrome C oxidase is an enzyme that is useful because it takes electrons off of cytochrome C in the electron transport chain and puts them onto oxygen. So you might then assume anybody who's got the ability to do aerobic respiration will be positive for cytochrome C oxidase. But as it happens, there's other enzymes with the same ability with different names. This is just one specific version. So again, we can't be really sure who's got it. We can be sure who doesn't. Can we say who doesn't have cytochrome C oxidase? Who can we be like 100% sure does not have cytochrome C oxidase? Yeah, everybody's quite confident that this guy who doesn't grow in oxygen doesn't have cytochrome C oxidase. That's true. There's another one that doesn't have it. Even though aerotolerant guys grow in oxygen, do they use oxygen for their metabolism? They don't. So they're not going to have any enzymes that would actually be part of respiration. It's likely, but not necessarily the case that cytochrome C oxidase is in these guys. They are doing aerobic respiration, but there's actually enzymes with the same function but different names, slightly different structure, basically doing the same thing. And this is, again, this is useful because the oxidase test can distinguish groups of organisms from each other, and sometimes even species from each other. So many of you will do an oxidase test as part of your unknown ID process as well. Last but not least, we've talked about nitrate reductase. In what process is nitrate reductase involved? Put that into the chat. When you go to reduce nitrate, what's the bigger picture? Why might you be doing that? What form of metabolism are you likely to be doing if you've got this enzyme? Yes or meat? That's right, Tiffany. So nitrate reductase is used in one of many possible forms of anaerobic respiration. Looking at these tubes, we can take a guess as to who might have this. Can we be sure? Can we say who doesn't have it for sure? Who does not have nitrate reductase of these tubes? There's just one. What is it? Yeah. We know that this guy is not doing any form of anaerobic metabolism, so it's going to be a no for this. These other guys are growing. They could be doing anaerobic respiration and they could be doing this form of it, but are they for sure? Can you tell? That's right, you can't. Okay. So now we're going to do some application questions to sort of try and prepare you for how we might use this information on an exam. So we've got two species, they're both mesophiles, and I'm only saying that so you know that their preference for temperature is the same. One of them is a facultative anaerobe, the other one is aerotolerant. We assume that they can grow in this broth that's being provided. So here are three broths where they've all been inoculated and grown in oxygen. There's three broths, there's only two species that have been inoculated. Okay, so one of these tubes is a distractor. A has a medium amount of growth, B has a lot of growth, and C has no growth at all. Which of these tubes represents the facultative anaerobe? Put your guess or your answer into the chat. One of these is aerotolerant. One of them is facultative anaerobe, and one of them is neither. It's not, it's just a distracting tube. And all of them were grown in oxygen. All right, so everybody confidently guessed A. That is not correct. Don't you feel good being in like good company with everyone? I feel like that's much more kind, like that's much nicer when you're like, I'm wrong, but I'm not alone in being wrong. Okay, well, let's think about it. If you are aerotolerant and you're grown in oxygen, what kind of metabolism are you doing? Are you doing aerobic respiration or are you doing anaerobic metabolism? If you're aerotolerant, grown in oxygen, are you doing aerobic respiration or are you doing anaerobic metabolism? Which one are you doing? Yeah, you're doing something anaerobic. The definition of an aerotolerant organism is one that doesn't use oxygen. It can survive. It doesn't care about oxygen particularly. It's got a way of generating energy that does not rely on oxygen, and it just does that all day long. Whether oxygen's there or not there doesn't really matter. If you're a facultative anaerobe grown in oxygen, what are you doing? Yeah, you're doing aerobic respiration. Which gives you more energy per glucose? Aerobic respiration or anaerobic metabolism? Which gives you more energy, aerobic respiration or anaerobic metabolism? Where do you get more ATP per glucose? Let's think about what we talked about earlier today. So fermentation, we know, gives you how many ATPs per glucose? That's one form of anaerobic metabolism. Some of these guys could be doing that. It gives you 2, okay. Aerobic respiration gives you how many? In a prokaryotic cell, Meliana. Sorry, in a prokaryotic cell. 38. And anaerobic respiration compared to aerobic respiration, anaerobic respiration being the other form of anaerobic metabolism, gives you the same or less? Yes. Anaerobic respiration gives you less ATP than aerobic respiration. We also know that fermentation gives you less ATP than aerobic respiration. So if we compare a facultative anaerobe that is definitely doing aerobic respiration in oxygen to an aerotolerant organism that is definitely not doing aerobic respiration, who is going to have more ATP and grow faster? The facultative organism. So which tube represents the facultative organism? Growing oxygen. Great. Does that make sense? Yes, no, or kinda? The answer that many people are giving now is B. Your first answer was A, and that was incorrect. Now everyone's saying B. Does that make sense? Great. So we have yes, no, and kinda. Perfect. So don't fret about it too much. We'll revisit this in a second, but this is an example of a question similar to what you might encounter on the next exam. All right, let's do another one. So now there's 3 options, right? We have no growth, medium growth, and lots of growth. But this time we have 2 tubes that are inoculated with the same guy, the facultative anaerobe, grown at its favorite temperature for 8 hours. One tube was grown with air and one without air. Which of these tubes represents the tube grown with no air? So in this situation, we're not talking about 2 different organisms now. We're talking about a facultative anaerobe inoculated into 2 identical tubes, but one's grown in air and one's grown without air. Which of these 3 pictures best represents the one grown with no air? Great. Everybody's guessing A, and this time A is the right answer. If you understand why A is the right answer and can defend your answer, don't make me cold call you, just raise your hand. I will cold call you if necessary. But go ahead and raise your hand if you can explain, like, why did you know right away that it was A? Sarah, go ahead.
I guessed A because it looks like there's less growth in that tube, and since facultatives prefer oxygen, it looks like there's a lot more growth in B. So yeah.
We know it's not C because this is a facultative anaerobe, and even if it's grown with no air, we know it's going to be alive, so it can't be C. Now we have these 2 options, but we know that a facultative anaerobe grown in oxygen would grow a lot, and grown without oxygen grows less. So this one makes more sense. But again, I want to come back to this, right? I think that, let me see if I can back up my slides to here. pretty much we all recognize now, we've kind of all internalized what this is. When we see this pattern of growth, lots at the top, growth throughout the tube, but the rest of the tube, there's less growth. What do we call this category? Put it into the chat. What do we call this category of organism that grows well at the top near oxygen and can grow without oxygen, but just grows slower? Yeah, this is a facultative anaerobe. And the definition of a facultative anaerobe is it prefers oxygen, which is really kind of a bit of a misleading term. It's going to grow faster in oxygen. And it has a way to grow without oxygen, but it's going to grow slower without oxygen. What is it doing here that makes it go faster that it's not doing here where it is slower? What is it doing at the top? What metabolic process is it doing at the top? Yeah, it's doing aerobic respiration. And down here, it's doing something that's not. We don't know what it is. Is it fermentation? Is it anaerobic respiration? We don't know. We just know that it's not aerobic respiration. And so you just know, because I've told you, because I've tried to explain why, but also from this picture, that aerobic respiration does in fact give you more ATP per glucose. That is why it is growing faster. It's not because it's like happier per se. It's just like legitimately, logistically, every glucose that's put through aerobic respiration, you get 38 ATPs. And when you try to use any other strategy to get energy out of glucose, you just get less ATP. So we're going to grow faster when there's oxygen. If you can do aerobic respiration, you're going to grow faster in air. Okay. So let's briefly touch base about the unknown ID project as it relates to what we've just talked about. As I said, for your unknown ID project, you're going to do a variety of tests, most of which are metabolic or growth tests. And we have all of the following things that are what we've talked about and more. So we've got the ability to grow your unknown at various temperatures. So what's its temperature preference? Is it growing well, medium, or poorly, or not at all? We have tests that measure its ability to survive at different pHs and different levels of salt. And so you just order a broth at 4 degrees or a broth at pH of 9 or a broth at 10% salt. If you want to understand whether or not your organism can grow in oxygen, whether it needs oxygen, is it able to grow without oxygen, you would do a thioglycolate test. If you want to know whether or not it has the enzyme catalase or the enzyme cytochrome C oxidase, these are tests that you can ask for. We have a nitrate reduction test, which is called the nitrate test. We have a sulfate reduction test called Kligger's iron auger. We have all sorts of exoenzyme tests, from a starch plate to a milk plate to a lipase plate. And we've got all kinds of fermentation tests. So these phenol red broths that you inoculated last week and that you'll look at tomorrow, test for, can you ferment this sugar? How about this one? How about that one? How about this one? So that's right. The more ATP per glucose, the faster you can grow. That's exactly right. Let me just show you some examples of these tests. You're going to get a chance to see this yourself in the lab, but I want to give you a preview and a review and a reinforcement, and also ask you a question that might show up on an exam. So here are 2 slides. The catalase test was performed on both of them. You'll get a chance to do this in class tomorrow in the lab, but what's been done is the cells of organism A and the cells of organism B have been applied to the glass as a dry smear, and then the hydrogen peroxide was added on top. Catalase test tests for the presence of the catalase enzyme. in these cells. And catalase uses hydrogen peroxide and makes oxygen bubbles in water. So which of these has the catalase enzyme in it, A or B? Right. Obviously A does and B does not. Now, I cautioned you earlier against over-interpreting the catalase test. If this is what you saw, you wouldn't necessarily be able to say with confidence that this guy does not do aerobic respiration, because there's a different enzyme that can substitute for catalase. But you can be sure that this guy does aerobic respiration, because only guys that do aerobic respiration have catalase. But if I told you that one of these organisms was a facultative anaerobe and the other one was an obligate anaerobe, which one would be A? Good. Yeah. Obviously, if you knew that there's only two options and you knew what those two options were, you'd be able to distinguish them. Clearly, if there's only two options, a facultative anaerobe, which is able to do aerobic respiration, and an obligate anaerobe, which is killed by oxygen, therefore has no defenses, then you know which one's which. Similarly, the oxidase test is done as a dry smear. It's done onto paper that's soaked with the chemical that the cytochrome C oxidase enzyme can react with. When it does, it turns purple. So you just apply your cells that might or might not have oxidase enzyme onto the substrate and see if it turns purple. So if you see this result, and let's suppose that this result on the left is what you get when you do your unknown, what can you say? If you see this purple color after you ask for the oxidase test for your unknown, what does it tell you? Yeah, it's got cytochrome C oxidase. Furthermore, and so that's the most basic and important thing you can say. You also can infer that it does have the ability to do aerobic respiration. You do have to be a bit careful. If you get a negative, it means it doesn't have cytochrome C oxidase, but does not necessarily mean it can't do aerobic respiration, because like I said, cytochrome C oxidase is one of the type of the enzymes that can do that step in aerobic respiration, but there are alternatives in other microbes. So they might be able to do aerobic respiration just using a different enzyme. Okay, another one that you're going to do in the lab tomorrow, nitrate test, nitrate reductase test, checking to see if nitrate has been reduced. All I want to say about that right now is that this is a two-step test where if it turns red in the first step after the first two reagents are applied, and those are sometimes called nitrate 1 and nitrate 2, also sometimes called sulfonilic acid and alpha methylamine, also called sulfonilic acid and VPA. You'll see those in your instructions tomorrow. But if it turns red in that first step, that means that it has reduced nitrate to nitrite. And then if it doesn't turn red in the first step, you have to then do a second step to find out if it is, if it's not positive in this first step because nitrite was further reduced or if nitrate was never reduced. And so as it turns out, red after the second step, which is the application of zinc powder, means that there's nitrate in the tube, which has never been altered. And clear or slightly gray after the addition of zinc means that nitrate is all gone and it's been turned into something that's not nitrate. So we'll go over this tomorrow. That's just an introduction. It's confusing at first and then it becomes much easier to understand. So based on what I've said, are you able to kind of give me a, I think I understood at least what you told me the different colors mean. which of these tubes is or are positive for nitrate reduction? So let's call this 1, 2, 3, and 4. So which one or ones would you say? And again, don't worry about being right or wrong. Just see if you can get there. It's uncomfortable to put a guess in and be wrong, but it's really, really important that you get used to that and be okay with it, because that is what learning is, right? And if you can't do that and you are afraid of that, it's going to hold you back. So I appreciate all your answers. It's fine to put question marks after them if you're like feeling kind of unsure. Good job, everyone. Okay. So I will tell you, and then you will be like, what? And then tomorrow, I hope it makes sense. So 2, 3, and 4, all positive. 1 is negative. Isn't that fun? Red is negative if it happens in step 2, and positive if it happens in step 1, and clear is positive if it happens in step 2. So next class in lab, we'll talk about this. I just want to prepare you for that, okay? All right. Last few things. Remember earlier today, we talked about exoenzymes, these enzymes that microbes secrete into their environment in order to degrade macromolecules. For example, this is a milk plate. In it, there is shockingly milk. Milk is opaque because of an enzyme called casein, not an enzyme, a protein that's slightly insoluble called casein. If an organism has the exoenzyme caseinase, then it will grow on the milk plate and secrete caseinase into its environment, and then it will clear the milk. This plate has 2 organisms. They both grew. Which one has caseinase, left or right? That's right, the one on the right. And this is an important thing that bacteria have, one of many different types of exoenzymes, because they don't have a GI tract. This image I stole from the internet, and it's actually reversed. So this is a lipase plate. This is a dye that's lipid-based. These organisms have both grown on it. The one that can digest the lipid-based dye by secreting lipase into its environment is clearly the one on the bottom. Here is the starch plate. Starch plates have starch in them. Both these organisms have grown. After they grew, you were able to determine whether or not these exoenzyme that digests starch was secreted into the environment by adding iodine, which binds to starch and turns it black. So which of these has the exoenzyme to digest starch, left or right? Remembering that if you digest starch, you clear the starch. So everywhere that's dark, there's still starch. Here, there is not starch. Good. And what was that enzyme? Correct. It was amylase. Okay. So a reminder that you're on this pathway. You should be getting close to, if not already finished, step 2. Stick around to hear questions about any of that. And you want to move on to step 3 as soon as you can. And that's going to be as soon as I've graded your step 2 to say, yes, you're on the right track. Just a reminder of what's due. Of course, tomorrow we meet for lab. And then this weekend we have the metabolism review quiz, and lab 5 is due, and lots of other fun things, including your unknown, making some progress there. If you have no questions, go ahead, log out, enjoy the rest of your night, come to lab ready to reinforce the things that you need. And if you have questions, please stick around and let's get them answered.